In this micro lecture, I'll be reviewing our chapter 8 on cellular respiration. So cellular respiration is one of the processes by which we are able to generate energy in the form of ATP, adenosine triphosphate. So we said ATP is the energy currency of our cell. So in this lecture, we will take our time and look at the cellular respiration process, the general overview in this process. And because the mitochondria is the main powerhouse organelle of the cell, we'll look at how it's really critical in the cellular respiration process. We'll look at other processes such as fermentation and how this occurs in the absence of oxygen within the cell. And we'll look also in general for the metabolic process, how we are either generating ATPs through the breakdown of molecules in catabolism, or how we are using that ATP molecule to build molecules in our body, such as proteins, the carbohydrates, the nucleic acids that we need. So first of all, we all eat various food. It could be breakfast, it could be lunch, it could be dinner. But what happened to the lunch that you ate? This lunch has various food components. So there's carbohydrate in there, there's proteins, there's lipids, there's minerals, there's vitamins. What happened to all these molecules in the lunch that we need? So through the process of cellular respiration, which involves the breakdown of carbohydrates, we are able to convert these molecules into ATP, which is adenosine triphosphate. These carbohydrates are synthesized from photosynthetic organisms like plants, and we are able to use photosynthetic byproducts as reactant molecules in cellular respiration. So during cellular respiration, oxygen, which is produced during photosynthesis, is consumed during cellular respiration. And because it's using oxygen air, this process is said to be aerobic. And it will result in the production of carbon dioxide gas and also water in the process. Cellular respiration does not occur all at once. So the cell takes a gradual step-by-step approach to be able to efficiently produce energy in the form of ATP so that the cell can use it. Now, if this process occurs spontaneously or all at once, the cell would be able to have a lot of non-usable energy in the form of heat, which will not make the process efficient. Now, since this is a metabolic process and it involves oxidation and reduction, so remember oxidation and reduction from our previous lecture, where oxidation involves the loss of electrons and reduction involves the gain of electrons, we need key enzymes that are oxidative and reductive enzymes that can help us in this particular metabolic process. Two of these enzymes is NAD+ and FAD. So we'll explore what is their role in the cellular respiration process. Now, this image right here kind of summarizes what happens during photosynthesis and how the products from photosynthesis are used in the cellular respiration process, and how these two processes are interdependent on each other. Now, as we are focusing on cellular respiration, and that pertains to the breakdown of glucose molecules, we could summarize the process in this equation. So cellular respiration involves the breakdown of glucose molecule, and you also are consuming oxygen in the process. And this is going to result in two key products, carbon dioxide and water, whilst releasing energy for us. Now, oxidation and reduction are really essential in the metabolic process. And in this cellular respiration process, glucose is what is oxidized. So glucose is able to lose some of its electrons, and those electrons are accepted by oxygen. So oxygen, which is gaining this electron, is reduced in the process. And this allows us to generate energy in the form of ATP through the cellular respiration process. Now, a critical look at the two key enzymes, NAD+ and FAD. So these enzymes catabolize these two metabolic reactions and help to speed it up. So nicotinamide adenine dinucleotide, NAD+, this enzyme is very relevant in oxidation and reduction. It can accept electrons and become reduced. It can give up electrons and become oxidized. Now, in the cellular respiration process, NAD plus would accept 2 electrons and a hydrogen ion, and it's going to become reduced to form NADH. So as we begin to break down the cellular respiration process, you would see NADH as one of the end products that we'll see in some of the steps. Another enzyme is going to be FAD, which is flavin adenine dinucleotide. This enzyme is sometimes used in the stead of NAD+, and is also capable of accepting 2 electrons and a hydrogen ions and become reduced to form FADH2. So as we go through the phases of the cellular step process in the cellular respiration process, we would see NADH and FADH2 as byproducts that is being produced. So again, I need you to think of the big picture. The food that we eat, the different molecules, the carbohydrates in it, how does our cell harvest this energy in the form of ATP? So the cellular respiration process is a stepwise process, and it includes 4 main phases. The first phase is called glycolysis. Glyco means sugar, to lysis means to break. So glycolysis simply means breaking down the sugar, which is in this case glucose. So during glycolysis process, we are able to break down the glucose molecule to generate 2 molecules of pyruvate, which is a 3-carbon molecule. So as we discuss each of these phases, I want you to think about which part of the cell is this process happening? What is the starting reactant molecule that it is using? What is the final end product that we are getting from each of these phases? Now, for glycolysis, it does not utilize oxygen. So in the absence of oxygen, glycolysis will still be able to break down glucose to give us 2 molecules of pyruvate. Now, the entire process of glycolysis is taking place within the cytoplasm of the cell. Now, the second phase is going to be the preparatory reaction phase. And during the preparatory reaction phase, we are able to change the pyruvate molecule, which was the byproduct from glycolysis, to acetyl-CoA, which can now be taken to the next phase of the cellular respiration process. So as we convert pyruvate to acetyl-CoA molecule, we also produce NADH, because the NADH plus enzyme is going to help us in that preparatory reaction phase, and it's going to become reduced to form NADH. And we would also have 2 molecules of carbon dioxide also being produced as we do this conversion from pyruvate to acetyl-CoA. The third phase is going to be the citric acid cycle, and this is happening within the matrix of the mitochondria. Now, the citric acid cycle is going to take acetyl-CoA, combine it with oxalic acetate, which is inside of the mitochondria matrix, to produce citric acid. And this will go through a chain of 8 enzymatic steps to give us 4 main byproducts. You're going to get 2 ATPs, 2 FADH2, 4 molecules of carbon dioxide, and 6 molecules of NADH being produced. Now, when you take 1 molecule of glucose, you get 2 pyruvate being produced during glycolysis. So the citric acid cycle is going to go twice. It's going to take 1 pyruvate at a time and give you a byproduct, and you're going to take another pyruvate and also give you another byproduct through that. Now, the last phase in the cellular respiration process is electron transport chain, ETC. This is a series of electron carrier molecules on the cristae, which is the inner foldings of the mitochondria. So electrons that are going to be collected from NAD and FADH2 are going to be passed to a series of electron carriers, and oxygen is going to be the final electron acceptor, which will help us to generate 32 to 34 ATPs during the electron transport chain, depending on the type of organisms. So for animal cells, they are able to generate 32 ATPs during the electron transport chain process. So let's look a little bit into the schematics. So remember, we want to break down glucose, and our big idea is that we want to be able to release energy in the form of ATP. So if you have 1 glucose molecule, it's first going to go through glycolysis. Glyco means sugar. So we're going to break the glucose sugar molecule to give us pyruvate. Everything during glycolysis is in 2, so you're going to end up with 2 ATPs. So you're going to end up with 2 NADH and also 2 molecules of pyruvate. Pyruvate is an energy-rich carbon molecule, so we can take it and then process it through the preparatory reaction phase, and then also go through the citric acid cycle, and then lastly through the electron transport chain. All those 3 reactions are happening in the mitochondria, which is the energy organelle inside of the cell, to help us generate a lot of ATP energy for the use for various cellular activity. So now let's take a little bit detailed dive into each of these phases. So with glycolysis, you are starting with the 6-carbon sugar glucose as your starting reactant. And you're going to need the help of the NAD enzyme to be able to come in and help us to be able to harvest the electron energy in glucose molecules. So NAD is going to be reduced in the process to become NADH, and glucose is going to be oxidized and is going to give us an output of 2 molecules of 3-carbon pyruvate. And before we can break down glucose, during the glycolysis process, we need to invest energy to energize the glucose molecule so that we can be able to split it or divide it or break it down. So you energize glucose with 2 ATPs and you end up producing 4 ATPs. But when you think about the net energy of ATP, you end up with 2 ATP as a net gain produced during glycolysis process. So in the glycolysis process, we are first investing 2 energies, and that is going to help us to split the glucose molecule to produce glyceride 3-phosphate. And with the glyceride 3-phosphate, we can go through a substrate level synthesis where, with the help of NAD+ enzymes, which become reduced, we are able to harvest electron energy from that. And the oxidation and reduction process help us to generate pyruvate as a byproduct during glycolysis. Now, in the second phase, which is the preparatory phase, this is happening inside of the mitochondria. And with the preparatory phase, we are taking pyruvate as the end product from glycolysis to change it and convert it to 2-carbon acetyl-CoA. Now, as we convert to acetyl-CoA, we are also able to produce carbon dioxide, because as you take pyruvate, it is a 3-carbon molecule. So we can remove 1 carbon through the carbonylation to generate carbon dioxide. And through that process, we'll be able to generate the 2-carbon acetyl, which combines with coenzyme A to form acetyl coenzyme A. And this would be able to get into the next phase where we can break it further to reduce and release ATP molecules for our use. So during the preparatory reaction phase, you are starting with pyruvate as your starting material. And the main goal is to be able to prepare it and change it to acetyl-CoA form. And in the process of doing this, you end up producing 2 carbon dioxide and 2 NADH also in that process as well. So right here, just to have a clear picture of the cellular respiration and the phases that we are looking at. This is the cell and this is the mitochondria. Now the mitochondria has an outer membrane, it has an inner membrane. It has an inner folding, which is called the cristae, and it has an outer inner hollow space called the matrix. So the third phase, which is the citric acid cycle, happens within the matrix of the mitochondria, right within this region. And the last phase, which is the electron transport chain, takes place on the cristae in there. So now when you take the citric acid cycle, it's going to take the product from the preparatory reaction, which is the acetyl-CoA, and it's going to now begin to break it down further in the cellular respiration process with the main goal to release ATP energy for the cell to use. So the very first reaction that happens during the citric acid cycle, also called the Krebs cycle, is that acetyl-CoA, which is a 2-carbon molecule, is going to combine with oxaloacetate, which is a 4-carbon molecule inside of the mitochondria. Now, the combination of those two will result in the formation of a 6-carbon molecule called citric acid. And the citric acid can now be further broken down through 8 enzymatic steps to generate for us 4 key end products. So you're going to have 6 molecules of NADH, 4 molecules of carbon dioxide, 2 molecules of FADH2, and just 2 ATPs being produced during the citric acid process. So when you think about the fact that we breathe in oxygen and we breathe out carbon dioxide, majority of the carbon dioxide that we are breathing out is actually coming from the citric acid cycle. So just a further look into what is happening during the citric acid cycle. So the product from the preparatory reaction, which is acetyl-CoA, would enter the matrix of the mitochondria, and acetyl-CoA would combine with oxaloacetate to now go through the citric acid cycle to generate for us those 4 key products. And these 4 key products that we see, so the 4 carbon dioxide, the 6 NADH, the 2 FADH2, and the 2 molecules of ATP, these are the main product that we're going to get from the citric acid cycle. Now, NADH and FHDH2 are electron carrier molecules, which we obtain as the coenzymes are reduced in these processes that we are going through. So we're going to take these 2 electron carrier molecules, which are rich with electrons, to the last phase, which is the electron transport chain. So when you think of the name electron transport chain, it simply means all the electrons that has been collected by the electron carriers, NADH and FHDH2, are going to pass it in a chain and get it to the final electron acceptor, which is oxygen in the process. So this whole process of electron transport chain is going to take place on the cristae of the mitochondria. And when you think about other organisms such as bacteria or microbes or other prokaryotes that do not have a mitochondria, the electron transport chain will still take place, but it's going to take place inside of their plasma membrane or their cell membrane. So during the electron transport chain, just like the name suggests, all the electrons that are being collected from NADH, and then you also have FADH2, they are going to pass all the electron energy to several electron carrier molecules on the cristae of the mitochondria. So you have electrons transported in the chain. There are various membrane proteins on the cristae that gets to pass on the electrons. So there are flavonone proteins, there are ubiquitinone proteins, there are metal-containing proteins, and there are also cytochrome proteins. These electron carriers pass all the electrons collected from NAD and FADH2 and pass it to the final electron acceptor, which is oxygen. So think about why we are all breathing in oxygen. That oxygen is really essential in metabolic process as it breaks down glucose in cellular respiration, because it is going to be the final electron acceptor to collect all the electron energy from NAD and FADH2. And then it can help generate for us ATP through oxidative phosphorylation as it pumps hydrogen and proton forces through ATP synthase enzyme to generate for us a lot of ATP energy for us to be able to use them in there. So just a quick recap. When you have one glucose molecule, you would be able to break these glucose molecule to release energy in the form of ATP as you go through glycolysis, as you go through the preparatory reaction, as you go through the citric acid cycle, and as you go lastly through the electron transport chain. Now, when you think about the 2 electron carrier molecules that we've talked about, NADH is able to deliver 3 ATP molecules, and FADH2 is able to generate 2 ATP molecules for us, even as they enter the electron transport chain process in there. So with the electron energy that has been collected by oxygen, it can pump hydrogen ions through ATP synthase, and this process is called chemiosmosis. So as chemiosmosis happens, we are able to get the major byproducts or end product from the electron transport chain, which is a total of 32 ATPs. So when you think about the fact that the glucose in our food, we get to break these molecules down to release ATP, and the ATP energy can help power the cell's activity. The electron transport chain is generating the majority or the most ATP that our cell needs. So just a quick recap. So during glycolysis, the breakdown of one glucose molecule resulted in 2 ATPs. The citric acid cycle gave us 2 ATPs, and the electron transport chain is giving us the majority, which is 32 ATPs. So in total, when you take one glucose molecule, you're going to end up with a total of about 36 ATPs being generated when you break down one glucose molecule in the food that we eat. Now, remember, we use thousands of ATP energy to power our cells' activity. So our cells generate way more ATPs for us to use for the various cellular activities that we need as well. And then also, when you think about these 36 ATP that our cell has produced, this is equivalent to about 686 kilocalories of the energy, only yielding 263 kilocalories of energy. So this 36 ATP is equivalent to 263 kilocalories, which is about just 39% of the energy in glucose. So remember when we talked about our lecture on energy, and we looked at the two laws of thermodynamics. So as we do energy conversion, as we break down the potential energy in our food to generate energy for us to use in the form of ATP, the second law says there's not going to be efficiency. So we're going to lose some as heat, and the efficiency as we break glucose molecules is only going to be 39%. We have the remaining 61% exiting out as heat in there. So again, the cellular respiration process is a stepwise process. It involves 4 phases, but the main big idea as we go through the 4 phases is to be able to generate energy in the form of ATP. We take one glucose molecule, it goes through glycolysis, the preparatory reaction, citric acid cycle, electron transport chain, and we end up with a total yield of 36 ATP being generated from there. Now, pyruvate, which is a byproduct from glycolysis, sometimes there may not be oxygen available, but we can still harvest energy and break pyruvate down. And the process by which we can break pyruvate down in the absence of oxygen is called fermentation. So in fermentation process, this is really straightforward. It's an anaerobic, so there's no oxygen involved. And it involves the help of NADH to help us transfer electrons to pyruvate and help us to generate various byproducts. So if you have yeast as microorganisms involved in the fermentation process where they are breaking down pyruvate, this is going to result in the production of alcohol and carbon dioxide. And this is what we use mostly in the production of alcoholic beverages, bread. So yeast fermentation of bread is also going to result in the production of some alcohol as well. Now, if you have bacteria and fungi going through the fermentation process, this is going to result in the production of lactate or lactic acid. And this is the main principle behind the use of industrial microbiology, whilst we are able to use these microbes to go through fermentation and produce cheese, we produce yogurt, and several other products in there as well. So fermentation is pretty straightforward. You have glucose molecule, which you can break it down through glycolysis to generate pyruvate. And during fermentation, where there's no oxygen, you can simply break down the pyruvate to get lactates, or you can get alcohol, depending on the type of microbe that is generating the fermentation byproduct for us. So just a quick look at if there's oxygen and if there's no oxygen. So if there's oxygen where you're going to aerobic respiration, one glucose molecule is going to give you 36 ATP. But in the absence of oxygen, we are able to have a quick burst of ATP through fermentation, and that will generate for you about 2 ATP molecules. And thus, we can get our muscles to be able to have some ATP energy to power its activity and for its use. But the main concern is that our muscles would also be exposed to other toxic forms such as lactate or alcohol, which are toxic to our cell. Sometimes lactate or lactic acid has the potential to change our cell's pH, and this may cause our muscles to be fatigued in the process as well. Another thing that yeast, which are key organisms that can go through alcoholic fermentation, when they produce these alcohol during the fermentation process, they can die in the process as well. And then lastly, fermentation is at a disadvantage because it has less efficiency of about just 2.1% ATP production compared to 39% energy production when you use aerobic cellular respiration in there. So remember, with fermentation, when there's no oxygen, you put in glucose, you get pyruvate, and you can convert the pyruvate to lactate or alcohol, depending on the type of microbe. So I'd like you to kind of have a brief summary of our lecture on the different phases of the cellular respiration, even as we want to break down one glucose molecule. So I want you to think about the glycolysis process. Where is it happening? Which reactant molecule are you starting with? And what is the final end product? Remember, our main goal is that we want to be able to break down glucose as we go through each of these phases and generate ATPs in the process. Thank you.